Hi, everybody. Welcome to week number 8, our final week together in Biology 120 Lab. Let's see what we've got going on. Well, as a recap from last week, you learned about the carrying capacity of ecosystems, and that's how many individuals can an ecosystem support without running out of resources, without building up too much waste, etc., to maintain the health of the ecosystem. This is terribly important. For example, in my state, we count how many deer are in different areas of the state, and then we allow hunters to take so many of those deer in order that we don't reach the carrying capacity of our ecosystems, because deer do graze a lot of plants and destroy a lot of vegetation. And they can overrun ecosystems, and that's just one example. We also learned about studying cohorts in a population. We do this, we talk about this all the time anyway. For example, we talk about baby boomers. I'm one of the baby boomers, and Generation Z, and there are lots of ways that we define cohorts in populations, especially these days, to talk about members in the same group. This week, you're going to learn about measuring population size, and this is tremendously important because if you're going to learn about the carrying capacity of ecosystems, you got to figure out how to accurately measure the population size of a species that you're interested in. And we're going to use 3 different methods. One is called the transect method, another is a quadrat, another is mark and recapture. And there are reasons to use these different counting techniques. And so you're going to have to figure that out. Why would you use the transect method over the mark and recapture method? What are the advantages of each? What are the different species that you would use these for? Okay, so you're going to have to answer examples of these different methods. And I have given you a reference here, your OpenStax Biology 2nd edition textbook, chapter 45. Got 1, so that is section 1 in chapter 45. This gives you specific examples of these population sizes and a species that you would use these different counting techniques for. So go to that chapter and look, the answers are right there. Some hints for the lab this week. You can use that wax pencil that you have been using all term for the transect activity and just wipe it off of this laminated sheet with a paper towel. It works perfectly fine. So go for it. Do it. You don't have to have a dry erase marker or anything. Just use that wax pencil. You already have it. And then you're going to figure out why one population counting method is better over another one for a different species, and that you will also explain in your examples. All right, a couple of last items for this term. All work has to be turned in by Sunday evening, Sunday, October the 25th at 11:59 p.m. When midnight rolls around on October 26th, the course is closed to you. You cannot add anything else. The course is also closed to me. So don't ask me to put anything into the course because I can't. There is no way to do this. The college closes the course except for grading. So the last day to turn in any work is Sunday, October 25th at 11:59 p.m. This is any unfinished work. Yes, you've been having grace periods of a week to turn in unfinished work, but there is a deadline for the term, and that deadline is this coming Sunday. So beware of that. You have to get any work turned in by that day and time. I would also ask that you please complete your course evaluation, especially the section about moi. Because my performance is partially based on these course evaluations, and it gives my team lead, who is an instructor like me, who leads a section of us, it gives my team lead and my dean an idea of how I'm doing and how the course is doing. You're also going to have input about the course and how we might change the course to make it better for upcoming students. So I ask you to please consider doing your course evaluation. It really helps me a lot. And of course, if you have any questions this week, if you're stuck on anything, there's my email address, m.sigmon@snhu.edu. Let me know, and I'll be glad to help you. Have a wonderful week, and I'll be talking to you this week to let you know how the grading's going. Have a great week.